Insulin is secreted by the which gland? Pancreas, right? So we'll see about their anatomy, external as well as internal anatomy. As you know, what is the location of pancreas? They located in the curve of duodenum, as well as this pancreas acts as a both endocrine as well as exocrine gland. How it looks? It looks like a flat organ which consists of three parts: head, body, tail. Now we're going to see the internal anatomy of the pancreatic gland. So if internally, if you are able to see, these are divided into two parts. One is called as acini, another one is called as a pancreatic islets. Acini is a part of the exocrine gland and the pancreatic islet is a part of endocrine gland. These acini, which acts as a part of endocrine gland, they are producing the digestive enzyme, and these digestive enzyme via the ducts it will enters into the GI tract for the digestion purpose. And in the diagram you are able to see in between these acini there are some cluster, and in that cluster you are able to see there are the various tiny cells are there, and this cluster is known as pancreatic islets. So if you will see the pancreas, the whole pancreas consists of 1 to 2 millions of tiny clusters, and these tiny clusters also known as islet of Langerhans, and these are producing the pancreatic hormones. Now we're going to see about the pancreatic islet hormones. As we know that there are the different types of cells present and these are secreting different types of hormones. So the cells are like alpha cells, beta cells, delta cell and F cells. So alpha cell carrying 70% of the pancreatic islet and secreting glucagon. The function of the glucagon to increase the blood glucose level. Beta cell, which consists of around 70% of the cell, secreting two hormones. One is called as insulin, another one is amylin. Insulin lower the blood glucose level, and amylin, which is inhibit the glucagon secretion. Because of that it will helps to the insulin, as well as it helps to delay the gastric emptying, as well as it acts as a satiety center. Next one is delta cell or D cell, which constitute about the 7%, which is called as somatostatin. And somatostatin have the control over both insulin and glucagon, which is released from the beta and alpha cells. Next one is F cell, which is consist of remaining portion other than alpha, beta and delta cell. It secretes the pancreatic polypeptide. It also have the regulatory role over the somatostatin secretion, as well as he performs the gallbladder contraction, secretion of digestive enzyme by the pancreas. So all these are the functions carried out by the pancreatic islets hormone. After seeing the pancreatic endocrine hormones, now we're going to start with the specific one, which is called as the insulin. Here we're going to see about the in detail insulin, like their structure. We're going to study about their secretion, their regulation, as well as we're going to study about their action, their mechanism of action. So all these things will be covered in the insulin part. So before starting with the structure part, we're going to see about some history about insulin. So who discovered the insulin? Insulin is discovered by the Banting and Best. These are the two scientists. In 1921, discovered the insulin. Ever you heard about the Sanger? Yes. So what is the role of Sanger? The Sanger who determined the insulin amino acid sequence when he did that in 1955. Okay. Now we'll come to the actual insulin structure part. As you know, insulin is 2-chain polypeptide having 51 amino acid. The A chain has 21, B chain has 30 amino acid. A and B chain held together by a 2 disulfide bond. This is the structure of insulin. But when they are synthesized, it is not in this form. Now we're going to see what are the different forms of insulin and how they are get synthesized and how they will form the insulin. So insulin is synthesized in the beta cell of pancreatic islets as a single chain peptide, which is called as a pre-proinsulin, having the 110 amino acid, from which 24 amino acids are first removed to produce the proinsulin. The connecting C peptide, which is of 35 amino acid, is split off later on by the proteolysis apparatus. So both insulin and C peptides are stored in the granules within the cells. So these granules within the beta cell store the insulin in the form of crystal of 2 atoms of zinc and the 6 molecule of insulin. So the granule of the beta cells, what they are having? They are having the zinc and insulin together, and with that C peptide is also there. Whenever there will be the secretion of this insulin, along with the insulin, C peptide is also secreted in the blood. The entire human pancreas contains about 8 mg of insulin. So if we are going to see the amount, 8 mg of insulin is produced by the pancreas, and it is also stored in the insulin, stored in the beta cells. Next we are going to talk about the regulation of insulin secretion. The main factor controlling the synthesis and secretion of insulin is the blood glucose concentration. Under the normal condition, 1 unit of insulin is secreted per hour by human pancreas. Whenever there is a meal, that time it will secrete much larger quantity of the insulin. The secretion of insulin from beta cell is regulated by chemical, hormonal and neuronal mechanism that we're going to see in the detail. Now we're going to see the chemical mechanism by which insulin release. So this is a beta pancreatic cell which have the ATP sensitive potassium channel on their surface, and this ATP sensitive potassium channel determine the resting membrane potential of the beta cells. So whenever glucose enters in the beta cell via the membrane transporter, which is called as a GLUT2, they will undergo the metabolism. Means glucose will undergo the metabolism via the enzyme which is known as glucokinase. This enzyme is very important, and this enzyme is also known as glucose sensor, because these glucose sensor will link the insulin secretion to the extracellular glucose. Means wherever there is an increase in the glucose level, the high glucose level will cause this glucose to be enter via the GLUT2 receptor into the beta cell. And once they will enter into the beta cell via the GLUT2 receptor, this enzyme will act on that glucose. And after that activation of this enzyme, there is a process which is called as a glycolysis, and via the glycolysis there is a production of ATP. So as the glucose will enter, increase in the ATP will start, and once this ATP will get increased, the ATP sensitive potassium channel will get blocked up due to the increase in the ATP, and this will cause the changes in the membrane potential. The changes in the membrane potential will cause the opening of the voltage dependent calcium channel, and that will lead to the calcium influx, and the resulting calcium influx will cause the trigger of the insulin secretion.